Chapter 22 focuses on the lymphatic system and the immune system. The lymphatic system is quite interesting because it is both a vascular system, which houses a good proportion at any one moment of the body's fluid volume, and also it's the highway, if you will, highway and byway of your immune system. All right? And this first slide here shows an artist's rendering of the lymphatic system. Now note the extensive lymphatic vessels running through all the tissues of the body that really parallel your blood vessels, especially your venous system. And note the lymphatic system consists not only of vessels but also lymphatic tissues, right, including your tonsils, your lymph nodes here, the spleen, which is perhaps the largest discrete lymphatic organ, the bone marrow from which the cells of your lymphatic system arise, the so-called lymphocytes, important cells of your immune system that travel in the lymph and can be found in the lymph nodes. Here we see the major vessel that returns lymph to the bloodstream, the thoracic duct here. So in the next few slides, we'll talk a little bit about the anatomy of the lymphatic system and then launch into a discussion of immunity. Once again, the lymphatic system consists of tissues, organs like the spleen, cells like lymphocytes that are responsible for, or at least partly responsible for, the body's defenses. Lymphocytes are extremely important. They are a component of your specific immune response that we'll talk about a little bit later. But they respond to most invading pathogens, especially those that escape the surveillance and initial defenses of your innate immunity, like the inflammatory response. Bacteria, viruses, fungi, and other organisms as well. Lymphocytes are also responsible for so-called immune surveillance for abnormal body cells, your innate natural cells in your body, for abnormalities, such as dedifferentiated cells or cells whose protein complement on the membrane surface is somehow changed. These cells of your immune system surveil the body and try to knock out these cells before they can cause problems. And of course, a prime example are cells that are undergoing malignant transformation from normal cells to tumor cells and then malignant cells. Also, any foreign protein that happens to be floating around in your body fluids, the most common example would be the toxins either from bacteria that produce them, some fungi, or even toxins from insect stings and things like that. Your immune system is responsible for detecting these abnormal proteins and disabling them and preventing them from doing further damage. Here's a more detailed rendering of the lymphatic system. Note the extensive and fine network of lymphatic vessels. Remember that the fluid role of your lymphatic system is a reclamation role. Recall our discussion about what happens at the capillary-venule interface and the fluid movement across capillary walls into the interstitium and back, right? The hydrostatic pressure of the blood entering the capillary drives fluid out of the capillary into the interstitium, and then the oncotic pressure of the protein remaining in the blood at the other end of the capillary as that blood flows into the venule helps to reclaim that fluid and keep it inside the blood vascular system. However, that reclamation role of the oncotic pressure and so forth is not perfect. And some fluid does remain in the interstitium, and it's the lymphatic system that is sort of the reclamation network, if you will, that picks up this excess interstitial fluid and returns it to the blood vascular system, mainly via the thoracic duct and the right lymphatic duct that enter into the subclavian veins and therefore return lymph fluid back to the bloodstream so that you don't lose a little bit of your blood volume every time your blood circulates through the capillary network of your body. Now what we see here, again, we see the extensive network of lymphatic vessels, and we also see the location of some of the major lymph nodes, the so-called cervical lymph nodes here, the lumbar lymph nodes. There are para-aortic nodes that are situated parallel to the aorta, the inguinal lymph nodes, and so forth. There are many more nodes that are depicted in this diagram, but these are the major ones, especially of clinical significance, that we examine for regional signs of infection and so forth. And we also see some of the other lymphoid organs and tissues, like the spleen. Right? The spleen is an interesting organ, as we know. It sort of doubles as a lymphatic organ and also a hematological organ, because it's the side of the spleen where red blood cells tend to congregate and die and release hemoglobin so that it can be broken down and part of it reclaimed and so forth. But it also acts as sort of a giant lymph node or lymphatic fortress where lymphocytes hang out waiting for foreign agents, whether they be pathogens and so forth, to pass their way so that these lymphocytes can meet that challenge. But in any case, we see that the lymphatic system it parallels the venous system in many ways, and again, that's part of its reclamation role. The lymph fluid from the lower extremity flows superiorly and collects here in the cisterna chyli, right? And the cisterna chyli will take the lymph from the lower extremities up through the thoracic duct, right? Okay, and the thoracic duct will empty the lymph back into the blood vascular system here, either via the left lymphatic duct, a branch off of the thoracic duct, or the right lymphatic duct. Both of these lymphatic ducts empty into the left and right subclavian veins, respectively, thus returning that lymph fluid to the blood vascular system and maintaining your blood volume. And the next 3 slides will review that information in textual form. And here we see a bit more detailed rendering of the things we've just talked about here. Note the cisterna chyli right here, all right? Gathers lymph from the lower extremities and transports it superiorly via the thoracic duct here. Right, and the thoracic duct, the left thoracic will branch into the left thoracic duct, which will empty most of the body's, or most of the lymph collected from most of the body into the left subclavian vein, and then the right side of the head and neck and the right upper extremity. That lymph will be emptied into the right subclavian vein via the right lymphatic duct here, right? And therefore the fluid, as I said, that's reclaimed by the lymphatic system will re-enter the blood vascular system via the venous system, and eventually back to the heart and back into the arterial system. Let us switch gears a bit now and talk about the particulars of the body's immune response. Right? The immune system, good grief, we could spend an entire year talking just about the immune system, and we probably still couldn't cover all of its complex aspects. But the immune response, we can say, developed or evolved over the millions of years of our own development as a way to protect our bodies, if you will, from the scavengers of nature, those pathogens and toxins and anything really in nature that would seek to take advantage of us and our nutrient-rich cell structure, right, for their own purposes, right, in order to defend ourselves and survive in a natural world that would just love to have us as a snack, so to speak. The immune system has evolved, and it has evolved to be an amazingly complex and adaptive system, right? And if you think about it, you know, all through your life, you've witnessed in a sort of a gross level your immune response, whether when you injure your knee and your knee swells, or you get a cold and your nose gets stuffy and you cough, to runny nose, you know, even when your skin gets red in a sunburn, or so forth, so on. All of these things are aspects of your immune response and your body's defenses being marshaled to meet some sort of threat. And, you know, if you think about it, the immune system is an almost unbeatable champion, right? There are only a few things out there that really overcome it, because all the colds and flu and bites and all sorts of things that your immune system has to respond to for most of your life, the immune system has defeated. The fact that you are sitting here listening to this lecture is evidence that your immune system has always won in the end, right? So it's an amazingly complex and adaptive system. And what you're looking at on this side is some of the characteristics of antibodies. And we'll talk about antibodies and where they come from. But antibodies are protein molecules. You see one here, one here, one here. This is a digital rendering of the crystalline structure of an antibody. An antibody is often drawn as a sort of a Y structure because its crystalline structure sort of mimics that here. We'll talk about the nature of antibodies, but we can say that antibodies have binding sites on them, all right? And these binding sites are specific for antigens, which are usually proteins or glycoproteins on the surface of some sort of foreign agent. Right? So antibodies develop binding sites that are specific for antigens, usually on the surface, not always, but usually on the surface of some sort of invader. It doesn't have to be a live invader or even a virus. It can be a protein toxin, right? But these antibodies bind specifically to these sites, and various things can happen as a consequence of this binding that results in disabling and elimination of the threat. But to talk about the immune system as a whole, we need to talk about both the specific component of the immune response, which involves lymphocytes and antibodies, as we've discussed here, but also the non-specific response, the so-called innate immune response, the response that's waiting, if you will, in the wings for any particular invader, any particular threat that it comes into contact with. And you can think of it this way. As I said, there is an innate sort of set of immune reactions that are waiting in the wings for any threat or any invader that comes along. And if the innate immune response, the non-specific, is sufficient to eliminate the threat will be eliminated, right? Often in a sort of subclinical, quote-unquote, way, meaning you may not even be aware of the fact that this battle was waged and won. However, in some cases, the innate response is not sufficient to eliminate the threat, and the specific or adaptive component of the immune response has to come into play. When that happens, usually, again, not always, but usually this is when the situation becomes clinical, meaning you feel symptoms, you exhibit signs of some sort of illness or injury. And during this time, the specific and adaptive arm of your immune response is literally developing a specific response to this particular threat in order to eliminate it. And that takes time, right? It takes time for the immune system to educate itself as to what is the nature of this particular threat, what do we need to build in order to eliminate it, and once the immune system figures that out, it sets upon building that response. And that takes a little bit of time, which is why you don't get over a cold or the flu immediately. It takes sometimes 10 days to 2 weeks or more, depending upon the nature of the threat, right? So we'll talk about the so-called innate or nonspecific response, and then we will talk about the specific and adaptive component of the immune response. So components of the nonspecific innate arm, if you will, of your immune system consists of physical barriers such as the integument, phagocytes, which are special cells that actually engulf or eat. In fact, the prefix phago means to eat other cells or even proteins. We'll talk about immunological surveillance, interferons, which actually are chemicals, the complement system, which is a network of proteins, the inflammatory response, and of course the fever response. All of these are aspects of the immune system's innate or non-specific defenses, and we'll take a look at each of these in greater detail in turn. All right, the importance of the physical barrier aspect of your immune system cannot be understated or underestimated. We see this in burn patients who lose aspects of their physical barrier because of burn injury, and we see the importance of the physical barrier aspect because these patients become susceptible to all sorts of infections, and often it is infection that causes these patients to succumb. But the physical barriers basically are just that. They keep hazardous things outside the body. The integument is perhaps the most visible aspect of the physical barrier, extremely important, especially the epithelium of the integument, the so-called epidermis, right, and secretions of the epithelium, the sebum, the sweat from sweat glands, all of these things, sort of prevent the approach and deny access to pathogens. But also epithelia throughout the body, the epithelia that lines the sort of inner chambers and inner cavities of the body. The epithelia deny access because they are selective in what they allow across them, right? Deny access to the deeper tissues of the body. Mucus, from mucus glands and goblet cells and epithelia is an example of a secretion that can trap foreign bodies. And these epithelia have cilia as well that can sweep the mucus away and sweep the foreign particles and pathogens away along with it, right? So again, the physical barrier function, the function of the physical barriers of the body cannot be understated. Extremely important. For that reason, the integument is often called an organ of the immune system. Part and parcel of the immune system because of its barrier function. Another very important aspect of your innate/nonspecific response is the so-called phagocytic response. Now, phagocytes are cells that take up debris or foreign particles, respond to any kind of invasion by a foreign agent, usually pathogens. These include the monocyte/macrophages. Monocytes are circulating cells of your immune system that circulate in your bloodstream and then migrate out of your blood vessels to the tissues where they mature into macrophages. Macrophages tend to be large multinucleate cells, not always, but often they are, and they produce lytic enzymes that attack a foreign agent once the agent is engulfed within the membrane of the macrophage. And that's what phagocytes do. They take up foreign particles, pathogens, and destroy them, and also take parts of the foreign agent that are left over after the destruction and present them on their cell membranes. And the reason they present them, and for this reason they're called antigen-presenting cells, is they are presenting antigens or or portions of the pathogen or foreign agent to the specific arm of the immune system to educate the specific arm of the immune system on what the nature of this pathogen or foreign agent is. And that education process informs the specific arm of the immune system when needed, informs it on how to design a response specific for that particular foreign agent. All right? Now, macrophages we talk about fixed ones and free ones. I mean, fixed macrophages are resident in a particular organ or tissue, like the Kupffer cells of the liver, the alveolar macrophages that we find in the lungs and so forth. But again, macrophages are cells that mature and differentiate from the monocyte that circulates in the bloodstream. Right? But it's not that monocytes die or divide and produce another cell. Literally, the monocyte will migrate out of the bloodstream and once into the interstitial tissue, will mature and differentiate a bit further into a phagocytic macrophage. And then there are the neutrophils and the eosinophils. The neutrophils are sometimes called the first responders of your innate immune response. They are the cell that tends to arrive first, and are also a major aspect of the inflammatory response. So when your body is facing an injury or a foreign invasion of any kind, the first responders are usually the neutrophils, which try to contain whatever damage or threat there is. If they can, fine and dandy. If they can't, then the other arms of both the innate and the adaptive specific immune responses are put into play. Now, we talk about diapedesis and chemotaxis. Diapedesis is the way that these cells actually move. If a monocyte, for instance, leaves the blood vessels and migrates into the tissues, how does it move to the site or get to the site of infection or injury? And it does so by a process called diapedesis. Now, diapedesis is really involves a cytoskeleton that is internal to the phagocyte, and we'll use macrophages as our example. Diapedesis consists of extensions of the cytoskeleton of the phagocyte that extends its membrane and causes the cell to kind of roll to its destination. Now, what we mean by this is that we can take a look here. Here we have a cell, right? Now, a cell that's undergoing diapedesis, the internal cytoskeleton will extend the membrane out, extend it even more, and such that the cell is now unstable and will roll, right? And it will continue extending this membrane out, and the cell will keep rolling and rolling and rolling until it reaches its destination, right? So that's diapedesis, and it's the way these phagocytes actually get to the site of infection or injury. Now, when we're talking about chemotaxis, what we're talking about is movement of these cells along a path in a direction governed by the release of certain chemicals called cytokines, lymphokines. These are chemicals that are secreted both by damaged tissues as well as cells of your immune system that are like breadcrumbs, if you will, that cause these phagocytes to follow a trail, all right, to the site of infection or injury. And that's how they know where to go, so to speak. And again, the way they get there is through this diapedetic or diapesis model, all right? And immunological surveillance. Again, this is surveillance by your immune system of your body's natural or normal cells that are part of your body's own cellular constituency, right? And in this surveillance is to look for abnormalities in these cells so that they can be taken care of and dealt with, usually by destruction, right? Now, lymphocytes play a role in this, but also an important arm of your non-specific. Of course, lymphocytes are part of your specific immune response, but an important part of your non-specific immune response is to surveil for abnormalities in your normal body cells. And the natural killer, or NK, that's what NK stands for, your natural killer cells are cells that share this responsibility. These cells travel throughout the body the bloodstream and the tissues and monitor your cells for changes. For instance, cells that are undergoing, as I said before, tumorous or malignant kind of changes, killer cells will destroy them before they can cause a problem. Now, some people think that natural killer cells are actually a class of lymphocyte, but recent research has challenged that assumption. But regardless, these cells, similar to lymphocytes, recognize changes in the cell surface markers that are on cells. And cells that have undergone genetic damage, genetic abnormality, will often reflect this abnormality in the complement of glycoproteins on their cell surface. Remember, these glycoproteins tend to be coded for by the very DNA that has been changed or damaged in some way. These cells will recognize these changes, and of course, anything that is in the body that has a foreign or non-self antigen on its surface, these cells will also recognize. Now, interferons are not cells, but rather chemicals made and released by cells. Interferons are a part of a class of such chemical molecules called cytokines, right? Cytokines are chemicals made by cells that are regulatory chemicals. They are released, and these chemicals travel far and wide to marshal the immune response, also regulate it. So the sort of chemical regulating molecules that are released by cells of your immune system. Right? Interferons, however, are small cytokines. They're protein-based cytokines that are often released by virally infected cells. These interferons trigger the production of antiviral proteins in other cells. In other words, these are proteins that can interfere with the binding of the virus with the cell. Remember, viruses are not living. Viruses are basically segments of DNA that have to attach to your own cells and make their way into the cell, where they hijack your DNA or hijack the infected cell's DNA and turn that cell into basically a virus-producing factory. Right? And so the cell that's infected produces many, many, many copies of the virus and then releases those new copies into your body fluids so that those new copies can go do it all over again. Right? So interferons can trigger the production of proteins that interfere with the ability of the virus to do this. Right? Interferons also interfere with the ability of the virus to replicate once inside an infected cell. And again, these interferons are part of a class of chemical messengers, chemical regulators called cytokines. Now, the complement system is actually quite interesting because this is a series of proteins made by the liver that is marshaled into action by a few triggers, really, but their role is really the destruction of cell by destroying cell walls. A few things can trigger them, as I said. One of them is the binding of antibody to antigen on the surface of foreign cells. The binding of antibody triggers complement, which is a series of 11 proteins that, when aggregated together in a certain way, literally attack the membrane of this invading cell and create pores in it, which ends up in the destruction of that pathogenic bacterium or other foreign agent. The complement system you can sort of think of as a swarm of bees, a swarm of bees that, when activated, can wreak havoc on an invading pathogen. The complement system also has a regulatory ability. It attracts phagocytes to the site of their activity. It can also stimulate them and make them more phagocytic. Right? They are also an inflammation or inflammatory promoting series of proteins. And again, their direct role is to literally puncture or create pores in the membrane of foreign pathogens and foreign cells and actually destroy them. Perhaps one of the most familiar aspects of your immune response, it's part of the response that we've probably all experienced at some point, and we can feel it, we can see it, and this is the inflammatory response. Right? Inflammation, in many ways, is really the first responsive process of your innate immune system. It's a response that is, quite frankly, responsible for many of the symptoms that we associate with illness or injury. But inflammation defined is a localized response, meaning localized to the site of infection or injury, that produces really 5 things. 4 I have listed here. One is swelling, which we can call tumor, right? We can call this tumor. And then there is redness, right? Redness. We can call that rubor. Our word ruby comes from that same root, right? Heat or calor. Calorie, our word from that comes from that same root. And pain or dolor, right? Dolor. Okay, it should be a B, right? And finally, some people put a fifth characteristic of inflammation, and that is dysfunction of the tissue, right? Dysfunction. All right. In any case, we've all experienced that to one extent or the other. Again, imagine an injured joint, injured knee, injured ankle. It swells, it gets red, it feels warm, and it hurts. And along with that becomes dysfunction of the joint, right? But inflammation can occur in any tissue. Inflammation in the lungs, fluid builds up in the lungs as part of the inflammatory response, and many of the symptoms of pneumonia, for instance, that we feel are really part and parcel of the inflammatory response, right? But the inflammatory response is important because I would say it probably prevents many infections that we are not aware of from ever arising to our awareness, from ever becoming clinical, right? Inflammation can sometimes repair injury, but usually a temporary repair, right? It isolates the injury, protects it, enough so that it does not spread to other tissues, right? Inflammation can slow the spread of pathogens, and it also marshals all of your other defenses as well, all right? One of the key players of your inflammatory response are the tissue mast cells. Mast cells are another cell population of your immune system. And you'll note here with this image of a mast cell here, you note these granules in here, these secretory granules. These granules are cytokines. Remember, they're chemical messengers that are released by the mast cell into the body fluids that summon and marshal other aspects of your immune response and cause certain local changes in your tissues. For instance, blood vessels in the tissue are dilated, which increases blood flow. Capillary permeability is increased so that more fluid can cross the capillary and into the tissue. Phagocytes become activated so that they can go do their thing, complement as well, right? A clotting reaction is triggered so that the region of the tissue is walled off so that the pathogen, for instance, cannot enter the bloodstream and transport itself to other parts of the body. The regional temperature is increased, which tends to increase the rate of certain biochemical reactions and also makes it an unfavorable site for pathogens to grow and thrive, right? And also these granules begin to warn the specific and adaptive arm of your immune system that something has gone wrong, right? So what happens is the blood vessels dilate and blood flow to the region increases. We call that hyperemia, right? Hyperemic, right? Okay, hyperemia, right? And then as capillary permeability increases, more of the fluid in the capillary can escape into the tissues. This is responsible largely for both the redness and the swelling aspect, as well as the heat, because remember the plasma component of the blood is mostly water, and water is the heat trough of the body, right? And the swelling, redness, and heat, coupled with some of the cytokines that are released, acts on local pain nerve endings to produce a feeling of pain, right? So the inflammatory response is really a host of responses that are the first responsive or first responders to infection or injury. And if inflammation can take care of the problem, then that will be as far as it goes. However, if not, the inflammatory response will, as I said, also send out cytokines to warn the specific arm of your immune response, namely the lymphocytes, the lymphocytic response, that there's a problem that needs their involvement, right? Now, the first cell to arrive during the process of inflammation, the first phagocyte to arrive, if you will, is the neutrophil, right? The neutrophil that we saw in the earlier slide. The neutrophil is really the main cell type of the inflammatory response. It is both phagocytic in the sense that it can engulf pathogens, but also it contains cytokines that, as I said, can activate and warn the specific arm of your immune system, right? But what kicks off the inflammation is often the mast cell in the tissues releasing these granules that summon the neutrophils and cause all of these other effects that we talked about. And here we see a diagrammatic version of the inflammatory process, right, which usually begins with some sort of tissue insult here. It can be infection, can be tissue damage, or both, right? It causes a chemical change in the interstitial fluid, right, that usually involves mast cells releasing their granules. And these granules contain both histamine and heparin, the effect of which is to dilate the local blood vessels and also to increase their permeability to fluid. As a result, the area of infection or injury becomes swollen, becomes red, becomes warm, and usually painful. Again, these are probably things we are all familiar with. At the same time, the clotting system is activated to create a clot to limit, if you will, exposure of whatever the insult is to the surrounding tissues. Right? At the same time, these granules send out cytokines that attract and activate phagocytes, especially neutrophils, right? When you think inflammatory response, I'd like you to think neutrophil, right? Very important response, very important cell type involved in the inflammatory response. And those neutrophils will begin to remove debris, begin to phagocyte tissue, pathogens, whatever needs to be done, and at the same time release cytokines that signal lymphocytes as part of the adaptive arm to come to the site if needed. Now, when we talk about fever, what we're talking about is really a systemic response that raises the core body temperature above about 37.2 degrees Celsius, or about 99 degrees Fahrenheit. Now, the clinical definition of fever is a bit different from that. If you were a patient in the hospital, we would say 100.4, above which we would start to worry. But generally, fever above 99 degrees, we start to think there's something wrong. Now, as it is a systemic response, it is mediated by the hypothalamus in the brain. The hypothalamus actually has a nucleus that serves as the body's thermostat, right? This thermostat, under normal circumstances, is set at about 37 degrees Celsius, what you might consider 98.6, 97.8, somewhere in there. Your body temperature actually rises and falls during the day normally, right? Pyrogens are chemicals that are released by certain cells of your inflammatory response, mainly. Neutrophils, again, are olsen neutrophils that release cytokines, mainly a cytokine that is called interleukin-1, right? Interleukin-1. And there's an interleukin-1 alpha, and if there's an interleukin-1 alpha, there's also a beta, right? So interleukin-1 alpha and beta are pyrogens, right? The word pyro comes from a Greek prefix that means fire, right? So we have one alpha and one beta, right? These pyrogens travel far and wide to the hypothalamus of the brain and tell the hypothalamus, hey, the body temperature, the core body temperature needs to be raised, right? And the degree to which it will be raised depends upon the chemical concentration of these pyrogens that reach the nucleus in the thalamus, right? Now, toxins, antigen-antibody complexes, pathogens themselves can act as pyrogens, but usually they stimulate the release of interleukin-1 from cells like neutrophils, macrophages, and even mast cells to some extent. So the hypothalamus is told that, hey, the new body temperature is now, say, 37.2 degrees Celsius or 99 Fahrenheit. The hypothalamus will institute or instigate an entire series of body reactions to bring that core body temperature up. Mainly, one of the main responses that accomplishes this is shivering. Now, what is shivering? Shivering is a constant series of muscle contractions, right? Muscle contractions that raise the core body temperature. Why? Because muscles are energy-demanding tissue. Muscles require a lot of ATP to contract. we know that all the sum total of all metabolic reactions, their largest yield is not energy, but rather heat. And that heat can be marshaled by your body to raise the core body temperature. So literally by contracting your muscles or shivering, you are increasing the metabolic rate of that tissue, which basically creates a bunch of heat that ends up in the heat reservoirs of the body, namely the body fluids, which will raise your core body temperature, right? So it is a systemic response mediated by the hypothalamus in the brain, but kicked off by the release of these chemical messengers called pyrogens, which themselves are usually founded at the site of infection or injury. So far we have talked a good bit about innate or non-specific forms of defense employed by your immune system. By non-specific, I mean the forms of the immune response are not adapted or specific to any particular pathogen or injury, and whatever immunity or innate immunity you have is present at birth and is genetically determined. Right? Specific immunity, meaning an immune response that is tailor-made or specific to a particular pathogen or a particular foreign agent, is acquired immunity. And it's achieved by exposure to an antigen, right? An antigen meaning usually a protein marker or glycoprotein marker on the surface of a bacterium or on the coat of a virus, or some molecule or marker that is recognizable by your immune system, right? That's what we can call an antigen, right? So exposure to antigens, right, educates the immune system to acquire a specific response against that particular pathogen, right? So we've talked a good bit about the non-specific responses, everything from the barrier function, the inflammatory response, the phagocytic response, etc. So now let's distinguish that with the specific, adaptive, and acquired arm of the immune system, right? Now this slide gives you a general overview of the different overall types of immunity. This is kind of a global view of the different types of immunity. Over here, on the side here, we can see our innate immunity, right? This is, again, genetically determined. This is what you have at birth. It does not require prior exposure of your immune system to an antigen, right? But we're going to focus now on the specific or adaptive or acquired immunity, the specific response here, that does require prior exposure. Now, there are two overall types of acquired immunity. One is passive and the other is active, right? Active immunity is produced by antibodies that are made and are developed in response to antigens, right? Meaning this is your immune response, okay? When the specific arm of your immune system is activated, a specific antibody response is created, right? And that response is known as an active response. Now, there are two types of active responses. There is that which is naturally acquired and that which is induced, right? Induced active immunity is when we exogenously induce that immune response. A vaccine is the prime example of induced active immunity. We directly administer an antigen to you, right, to stimulate your immune system to react to it, such that when that antigen reappears in an actual infectious form, your immune system will be ready for it, right? And then naturally acquired immunity is immunity that you acquire by exposure, from exposure to antigens in your environment. This occurs, for example, during childhood as part of your development, right? You're exposed to certain antigens in your environment, and you acquire immunity to those things certain things that you are naturally exposed to. Passive immunity is not an immune response to antigen, but rather a transfer of someone else's immune response to you. And specifically, we're talking about the transfer of antibodies from one host to another, right? And there are two types of passive immunity, right? There is induced passive immunity, which is just that, when we physically separate the antibodies out from one person's serum and transfer them to another's, right? Much of the current treatment against Ebola, for instance, is focused on induced passive immunity. It is isolating antibodies from people who have recovered from the disease and using those antibodies to treat people who still suffer from the disease, right? Now, natural passive immunity specifically refers to the transfer of the mother's antibodies across the placenta This is via immunoglobulin G, which we'll discuss in a minute. But this is a transfer of maternal antibodies to the fetus across the placenta or in breast milk after birth, right? Now, your immune response has certain important properties. Now, again, we're talking about the adaptive or acquired arm, the specific arm of your immune response, not the innate, right? The properties of your specific immune response are its specificity, right? It is activated by and responds to a specific antigen, right? So it's specific, right? And it's able to confront almost any antigen that's presented to it, right? The adaptive immune system also has memory. Okay, if you can imagine, the cells of your adaptive immunity are created as an army that goes after the invader, and when the invader is eradicated, a certain number of soldiers of that army stays around, right? As sort of a memory response. So if that particular invader ever shows up again, meaning if that particular antigen is ever presented to the immune system again, the immune system has soldiers on the ready to undergo mitosis and multiply to respond to that antigen before you even feel symptoms or display signs of clinical illness, right? Finally, tolerance, right? Like all aspects of your immune system, it must be able to distinguish self from non-self, right? A normal functioning immune system responds to foreign antigens but ignores your host, your self tissues. Now, as for the immune response as well, and again, we're talking about the specific arm of your immune response, right? It begins with an antigen that triggers the immune response. This antigen has most frequently been phagocytosed by one of the phagocytes, a neutrophil, a macrophage, and when that neutrophil or macrophage or other phagocyte has engulfed that invader and broken it apart and processed it, it takes one of its antigens and displays it, right, on its membrane, right? So the phagocyte will literally display the antigen on its membrane and present that antigen, right, in fact, it's called antigen presentation, will present that antigen to the cells of your specific immune response, mainly T cells and B cells, T lymphocytes and B lymphocytes, right? T stands for thymus because the T cells develop and mature in the thymus gland, mainly during childhood. The thymus gland involutes or becomes inactive after the age of about 9 or 10 or so, right? And then B cells. B originally stood for something called the bursa of Fabricius, which is found in chickens. We can think of it as standing for bone marrow because B lymphocytes develop and mature in the bone marrow, right? So T cells and B cells, T lymphocytes, B lymphocytes, they mean the same thing, right? So the general course is this. An antigen-presenting cell, mainly a phagocyte that is now presenting this antigen on the surface of its membrane, exposes T cells to this antigen. This activates these T cells, which go after other cells, mainly the invader that is presenting that antigen, and also stimulate B cells to make antibodies that are themselves specific for that antigen. And those antibodies are released by the B lymphocytes into the body fluids where they chase down and attack anything else that's presenting that antigen. And this slide is a graphical or bird's eye view of the specific immune response. Begins with an antigen that is engulfed by phagocytes. Those phagocytes process that antigen and present it on the surface of its membrane, and then present it to T cells, right? T cells. Specifically, we are talking about a type of T cell called the T helper cell, or T sub H, T helper cell. The T helper cell is the major general of the immune response, right? The TH cell, because the T helper cell releases cytokines that regulate much of the rest of the specific arm of the immune response, right? So it serves as kind of the general. And as an aside, the human immunodeficiency virus, or HIV, specifically goes after the T helper cell, right? And by destroying T helper cells, you're destroying the major regulator of the rest of your specific immune response, which is what ends up crippling the body's specific immune defenses, making the patient vulnerable to all sorts of things that he or she normally would not be vulnerable to, right? So, in essence, phagocytes activated, and antigen is presented to T helper cells. T helper cells release cytokines that activate other T cells. These are a different type of T cell called the cytotoxic T cell, or T sub C, right? Cytotoxic T cells themselves go after, right, the invader and kill them by releasing cytotoxic enzymes, disrupting the membrane. These are attacking T cells, right? Because of this particular response of the specific arm of your immune system is called the cell-mediated response, because cells themselves go after the invader. T helper cells, however, as I said, also release cytokines to activate B cells, or B lymphocytes, causing B lymphocytes to make antibodies. And again, antibodies are not cells, they are proteins, right? They are proteins that have the capacity to bind very specifically, right, to the antigen, okay? So the antigen-presenting cell will inform the T cell, T helper cell, to inform the B lymphocyte to make antibodies that are specific for that particular antigen, right? And because B cells themselves do not attack the antigen, only the antibodies do, we call this arm of the specific immune response antibody-mediated immunity. So we have cell-mediated involving T cells, and we have antibody-mediated involving antibodies made by B lymphocytes. And as I mentioned in the previous slides, the major types of T cells to be concerned about, again, the cytotoxic T cells. These are the T cells or T lymphocytes that themselves attack any foreign cell, right? They go after, attack, and destroy any foreign cell, right? The helper T cells, as I said, these T cells release a multitude of cytokines that serve as regulators of much of the rest of the specific immune response. They activate cytotoxic T cells, even other T helper cells, and as I said before, they activate B cells to make antibodies. They're sort of the regulator cell or the general of your specific immune response. And then there are also a few suppressor T cells that roam around, right? Every car needs a braking system, and the suppressor T cells are there to limit and inhibit the immune response so that it does not get out of hand and it stays modulated, right? If the immune response is too hyperactive, you can potentially damage, collateral tissues and not just the tissue, the cells that you want to go after. Now, as we've said, the specific arm of your immune response is activated by antigen presentation. And these antigens are usually presented by phagocytes. Sometimes they can be presented, in the case of viral infection, by cells actually infected by the virus, right? But the antigen must be presented to the specific arm of your immune system. And it's usually presented on the surface of either infected cells or phagocytes that have taken up invading pathogens and such. And the presentation occurs in an antigen-glycoprotein combination on the cell membrane. Now, the proteins that present this antigen are called MHC proteins. MHC stands for Major Histocompatibility Complex. Now, these complexes were so named, they were first discovered in mice. Their human equivalents, we often use MHC as a convention, but their human equivalents are actually known as human leukotropic antigens, or HLAs. But if you say MHC, HLA, everyone will know what you're talking about. So these are glycoproteins on the surface of phagocytes that present antigen to the specific arm of your immune system, right? T cells are then activated upon contact with this particular antigen, right? So you have an antigen-presenting cell, or APC, right? And we'll call it a phagocyte, has this glycoprotein on the surface of its membrane, right, the MHC, or HLA, right? I'll put HLA. and it will present an antigen, right, to a T cell, usually a T helper cell, right? The T helper cell also has a surface protein receptor complex. It's often called the CD4 complex, right? But it will associate with this antigen presenting cell, and this T cell will then be activated. I'm just drawing the nucleus in here. This T cell will then be activated and release cytokines that activate B cells and other T cells and so forth, so on. Now, two classes of these MHC, or major histocompatibility complex molecules, have been found. Right? Class 2 are the ones that we have been talking about. They are found on antigen-presenting cells presenting antigens to lymphocytes. Class 1 is actually on every cell that has a nucleus in your body has a class 1 MHC. Right? The class 1 MHC forms part of the glycocalyx that you learned about in the first semester of the course, right? The glycocalyx, if I can spell it right, okay? Now, the configuration of the class 1 MHC is genetically determined, as is the configuration of class 2. Now, what do I mean by configuration? I mean the amino acid structure of the protein, right? The MHC class 1 is how your immune system distinguishes self from non-self. Your immune system knows that your MHC1 molecules on your cells are part of yourself, and therefore they do not attack. Right? Class 2 MHC, as I said, is the molecule that presents antigen to the specific arm of your immune system, found mainly on phagocytes, which serve as antigens presenting cells. Now let's return to antibodies for a moment. Again, antibodies are produced by B lymphocytes, right? B lymphocytes, not T cells, not T lymphocytes, but B lymphocytes, right? Antibodies are not themselves cells. They are rather proteins produced by cells, produced by B lymphocytes, right? In response to activation, in response to antigen presentation to a T helper cell, which then releases cytokines which activate B lymphocytes to make specific antibodies, antibodies that have specific binding sites to specific antigens that correspond to antigens on the protein, on the surface of bacteria, et cetera, et cetera. Now, antibodies are extremely important molecules, and they have a diverse set of functions. One of their most obvious functions is to bind to antigens on, say, bacteria, viruses, and bacterial toxins that prevent them binding to healthy cells, right? So it's sort of gumming up the lock. If you put gum in the lock, the key cannot fit and won't unlock the lock. So by neutralizing viruses and bacterial toxins, they cannot bind and harm cells. Antibodies can also mediate the process of agglutination. When we see blood typing, and you see the agglutination response that you observed in lab. Antibodies were mediating that agglutination between the type A and type B and so forth antigens on the surface of those red blood cells. It was the antibodies that was cross-linking these cells to clump them together, and we call that antibody-mediated clumping agglutination, right? Antibody binding to antigens on foreign cells also can activate the complement system, that series of 11 proteins we talked about that is activated almost like a swarm of bees that can create pores in the membrane of an invading bacterium, for instance, and destroy it. Also, this process of opsonization, which is a very specific term in immunology, an antigen that is coated with antibodies, meaning antibodies that bind to antigens on the surface of the pathogen, can actually enhance the capacity of phagocytes to phagocytose these pathogens and destroy them, and also present their antigens on their MHC class 2 molecules to the specific arm of your immune system. Okay? Antibodies can also stimulate and promote the inflammatory process, right? And along with neutralization, can prevent viruses from adhering to the surface of cells that they wish to infect. Again, that's sort of gumming up the lock so the key cannot fit. Now, these are some of the major classes of antibodies, right? The Igs. Ig stands for immunoglobulin, right? So immunoglobulin. Immunoglobulin G is really the workhorse of your antibodies, right? About 80% of your antibodies circulating at any one moment are immunoglobulin G. Immunoglobulin G, that antibody provides broad resistance against many pathogens, viruses, bacteria, toxins, even fungi. IgG really are the workhorses of your antibody response. IgE, that is an antibody that is involved mainly in the allergic response. It's a local tissue-specific response that enhances the inflammatory response that we talked about. The release of fluid across capillary walls, hyperemia due to the dilation of capillaries in local tissues, pain and swelling, etc. Another antibody to be aware of is IgM here. IgM, it's actually the largest antibody. This antibody actually exists in a pentacluster, or a 5-antibody cluster that circulates in serum. And this is the first antibody secreted after B lymphocytes have been activated. And its levels decline in serum as IgG levels rise. So really, IgM is the immature response of your specific arm of your immune system, followed by the mature response involving IgG. IgA we find mainly at the sites of mucous membranes in glandular secretions. We find it in mucus, we find it in saliva, we find it in mother's milk, right? IgA is the antibody in your secretions for the most part, important for immunity at those body surfaces. And this slide goes into the different types of antibodies in more detail. So study this chart carefully to get a good idea of the role and structure of each of these classes of immunoglobulins. So this slide gives you a graphical overview of how the immune response might unfold against a bacterial versus a viral pathogen. Right? This is sort of a bird's eye view of the response and how the different bacterial and viral responses contrast with one another. So if you look here to the left side of the figure, we see the bacteria has invaded somewhere in the body. A macrophage, resident in the tissues, and again, macrophages mature from circulating monocytes in the blood, right, that migrate out of the blood into the tissues and mature into phagocytic macrophages, right? So a macrophage will encounter this bacterium, engulf it, destroy it, and present one of its antigens on that surface of that macrophage, and it will present it to a T cell. Right? So this antigen presentation activates cytotoxic T cells, which will directly go after that bacteria. Any bacteria that is presenting that antigen will be destroyed by cytotoxic T cells. At the same time, helper T cells will be activated, which will release a myriad of different cytokines, which will activate B cells to begin producing specific antibodies against the antigens on this bacterial pathogen. Right? And when B lymphocytes are activated in such a way they themselves mature to become something called plasma cells. These are B lymphocytes that have become activated that now secrete antigen-specific antibodies as plasma cells. And if you look over here to the right, you will see that the immune response to a viral infection is a bit more complex, right? Because remember, viruses are not living. They are simply packages of DNA that gain access to your cells and hijack the DNA in the nucleus of your cells to turn that cell into a virus factory, right? So viruses infect your own tissue cells. They get inside those cells, right? So going after those cells directly can be somewhat of a problem because they are literally hiding in the nucleus of your normal cells, right? One of the first things that happen in the viral infection of your normal cells is, your innate response. Those cells release interferons. Remember the interferons. They cause the production of proteins that will resist viral infection, will resist the virus's ability to integrate into the DNA of the nucleus and take over protein production to turn, as I said, your cell into a virus factory. So interferons resist these processes of viruses, right? At the same time, cells that are infected by this virus will end up presenting antigens specific to that virus on their cell membranes, right? And those antigens can be presented to T cells, right? Cytotoxic T cells can destroy those cells infected by viruses. Natural killer cells can also notice the change in that normal cell's MHC structure. Remember, natural killer cells survey your normal cells for changes, right? And a normal cell that is infected by a virus will display certain changes in its antigenic profile on its membrane, which is noticed by natural killer cells. Both the activation of cytotoxic T cells and natural killer cells will result in the destruction of cells that are infected by that virus. At the same time, phagocytes can directly uptake this virus and present it to T helper cells, which can activate B cells, mature them into plasma cells, right, down here, and those plasma cells can produce antibodies that are specific to viral antigens that can bind to those viruses and neutralize them by preventing them from being able to bind to normal host cells, right? So very similar, but some differences because the virus pathogenic profile, if you will, is a bit different from that of bacteria. This slide titled Hormones of the Immune System, really what we're talking about are cytokines. That might be the more specific name for them. These are the cytokines, right? These are the chemical mediators that stimulate and regulate the overall immune response, okay? There are the interleukins. We've already met one of the interleukins. That's interleukin 1, alpha and beta, but interleukin 1 is the major pyrogen released by neutrophils and other cells that can set your body's thermostat at a different level via the hypothalamus, right? Interleukins, there are many types of them, IL-1, interleukin 2, interleukin 4, 5, 6, 7, there are many of them, and they all have very specific functions in regulation of your immune response, many of which are beyond the course of our discussion. Were this a course in immunology, we could, as I said, spend an entire year on this subject alone. But in any sense, interleukins are produced by B and T lymphocytes and also by macrophages, right? They have many functions. They stimulate B cells to become plasma cells and make antibodies. They activate T helper cells, which themselves release different cytokines, right? They enhance the inflammatory response. They also can limit the immune response when necessary so that it does not become hyperactive and begin causing collateral damage to your normal tissue. We have already met the interferons, which are chemicals that are secreted by virally infected cells, which cause other cells that have not yet been infected to make proteins that confer upon those cells a manner of resistance to viral infection. It makes it more difficult for viruses to gain entry into the cell, makes it difficult for viral enzymes to cause integration of the viral DNA into the normal host cell DNA, et cetera, right? Tumor necrosis factor, or the TNFs, are a major cytokine. They were first discovered because, in the laboratory they slowed tumor growth and they actually killed tumor cells, and that's how it got its name. But TNF, TNF alpha, TNF beta, the tumor necrosis factors are also important signaling cytokines, right? The colony stimulating factors, or CSF. These are what cause plasma cells and T cells to multiply, to help build that army we're talking about when needed to go after a specific pathogen. Now, we call someone who has a normal functioning immune system immunologically competent, right? Someone who is immunologically competent has the ability to demonstrate a proportionate immune response upon antigenic exposure, right? That's what immune competence means, right? Fetuses become somewhat immune competent before they're ever born because they receive immunity from the mother across the placenta. Immunoglobulin G, right, IgG, is the only immunoglobulin that can cross placental membranes, right? And so immunoglobulin G, or IgG, is an antibody that is made by the mother's B lymphocytes, but transported across the placental membranes into the fetal circulation and confers some degree of immunity to the fetus before the fetus is ever born, all right? However, once the fetus becomes an infant, meaning after birth, the fetus's immune system develops upon exposure to different antigens. Now, the immune disorders fall under two broad headings. There are the autoimmune disorders. The prefix auto means self, right? An autobiography is a biography about myself, right? Very boring read. I wouldn't recommend it. Autoimmune disorders are therefore disorders that involve the immune response to your own self cells. The immune response mistakenly targets your self cells. And as I've said to you many times, if you had to define the immune system in one sentence, it would be the ability to distinguish self from non-self, right? And then there are the immunodeficiency diseases. These, as the term implies, are diseases which involve a lack of immune competency. Either the immune system does not develop properly from infancy forward, or is blocked or destroyed somehow, as most familiarly by human immunodeficiency viral infection, or by chemotherapy, or other processes that involve suppression of the bone marrow, for instance. Now, the term allergy is a very familiar term, but in immunology it has a very specific definition, right? An allergy is an inappropriate or excessive immune response to an antigen, right? In this context, we call them allergens, but they're basically antigens that cause an inappropriate or excessive immune response. There are many types of allergic responses. The one most familiar to everyone, the runny nose, the sneezing, the itching, watery eyes, that is called immediate hypersensitivity, right? Type 1. But there are others. There are cytotoxic reactions. There are the immune complex disorders. In fact, type 3 hypersensitivity is an allergenic disorder. That is what is involved in many types of glomerulonephritis, right? The deposition of these immune complexes at the glomerular capillary membrane, which affects filtration across the glomerulus into Bowman's capsule in the kidney, right? And then there is so-called delayed type hypersensitivity, right? Anaphylaxis is when there is a system-wide or body-wide circulation of allergen that causes a massive hypersensitivity response throughout the body that can actually be deadly by a mechanism we'll see in the next slide. Now, allergic or hypersensitivity reactions are really a two-exposure series of reactions, right? It requires a first exposure to an antigen or allergen, followed by a second or later exposure that actually produces the allergic response that we're all familiar with, right? The first exposure involves, like any other antigen, being taken up by a phagocyte, like a macrophage, which then presents that antigen to a T helper cell. T helper cell releases cytokines that activate B cells. B cells mature into plasma cells and make antibodies, but in this case, the antibodies that are made are not IgM or IgG, but rather immunoglobulin E. And these immunoglobulin E antibodies will then bind to mast cells that are resident in the tissue. And remember that mast cells have these granules inside them that can be released. These are cytokines and chemical mediators inside these mast cells. When they are released, they cause all sorts of tissue changes, including dilation of the blood vessels, increased capillary permeability so that fluid can cross capillary membranes into the interstitium, and so on and so forth. So these mast cells are now primed with IgE that is now specific for the allergen, right? In other words, these IgE molecules are specific for antigens presented by these allergens, so that a second or later exposure of this allergen will cause the IgE molecules on the mast cell to bind or cross-link with these allergens. So note here in this picture we have the mast cell, we have the specific IgE, antibodies on the surface of that mast cell binding to antigens on the allergen. Right? This cross-linking of allergen, IgE, and mast cell causes massive degranulation or release of these cytokines from the mast cell, which causes all sorts of changes that we've talked about. The histamines, the leukotrienes, and other mediators, they cause vasodilation, they cause bronchoconstriction, they cause capillaries to leak and fluid to escape from the blood vessels into the tissues, and so forth and so on. Now, when this happens locally, we sneeze, our eyes water, our skin itches. But if this process that we have just outlined happens on a body-wide or circulatory system-wide level, this response can become deadly, because as your blood vessels vasodilate, your blood pressure drops. As fluid is lost across capillary walls, blood volume drops and blood pressure drops. As bronchioles and bronchi constrict, your airways become more resistant to airflow. When this happens on a massive scale, your body can no longer maintain organ perfusion, blood pressure has dropped, and you're in what is called anaphylactic shock, right? You need immediate intervention or you will die, right? And histamines, corticosteroids, epinephrine, also these molecules have properties that can block different aspects of this response. And if this occurs, anaphylactic shock, you need all of these agents very, very quickly, or this can be deadly.